Okay, so now it's time to talk about our next class of molecule that we're going to run into, and these are the lipids. So one thing that is characteristic of all lipids is that they are hydrophobic, right? Hydro, water, phobic, fear. They don't like water, they don't mix well with water. There's a lot of diversity. There's a very important class of molecules. Again, as we talk about these different molecules, we're going to talk about their building blocks and things, their characteristics, but in your brain, kind of compare and contrast these to other groups. So we just talked about the carbohydrates and how they're used for fuel and for some structural and what they're, they're built of sugars. So think about that and see how lipids are going to be very different. Like I said, the unifying feature is that they're going to be hydrophobic. The reason that they're hydrophobic is that they're mostly built of hydrocarbons. And remember, hydrocarbon is just a hydrogen and a carbon. Remember, we talked about, what are the most common hydrocarbons that you're familiar with? Things like fossil fuels, right? Oil. And you know that oil and water don't mix. And so if you've got these lipids that are mostly hydrocarbon, they're also not going to mix well with water. But what's that tell you about the energy in lipids? You remember that hydrocarbons are very high energy molecules, right? They've got lots of bonds that hold lots of energy. Well, if lipids are mostly hydrocarbon, then lipids too are going to be high energy molecules. So they have a very high energy density. Within the lipids, we have three broad categories that are important in biology: the fats, the phospholipids, and the steroids. So let's talk about fats first. So to make a fat, you've got a couple different monomers. You've got a couple different building blocks. You've got a glycerol and then some fatty acids, okay? A glycerol is just a 3-carbon molecule, a very simple molecule. And those fatty acids are attached to each of the 3 carbons. And so that makes you've got a glycerol that's got 3 fatty acid tails, a triglycerol or a triglyceride. And that maybe is a term you've heard before. So if you hear triglyceride, you automatically think fat, automatically think lipid. So those fatty acid tails that come off the glycerol are mostly hydrocarbon. And so again, this is why these fats have lots of energy. So this is a diagram of a typical fat. And you can see in the gray on the left there, you can see the glycerol, 3 carbons. And then coming off of each of those carbons is a long fatty acid. And that fatty acid is mostly a hydrocarbon. And so why do they call them fatty acids? Technically, that functional group that connects them to the glycerol is called a carboxylic acid. These are things that you'll learn more in organic chemistry. But you can recognize the 3 tails, and those tails are mostly hydrocarbons. So there's lots of energy here. And that's kind of what I'm getting at with this slide. What do we know about hydrocarbons? They have lots of energy. Fats have 3 hydrocarbons, so they have lots of energy in a small space. Now, we said that plants mostly store their energy in the form of carbohydrate or starch. Animals tend to store their energy mostly as fats. And so for short-term energy storage, sometimes we use carbohydrates. Remember glycogen? So that's for kind of short-term energy storage. But if you have extra energy in the long term, you put it on as fat. Fats store a ton of energy in a small volume, so they have a very high energy density. And when we talk about fats, there are some terms that you're probably familiar with. Saturated fats versus unsaturated fats. Saturated fats tend to be solid at room temperature, and unsaturated fats tend to be liquid at room temperature. Now, if you look at each of these examples, you can see for both the saturated and the unsaturated fat, if you look closely, you can see the 3 carbons You can see the glycerol, and then you can see the 3 tails coming off of each of those carbon, and those tails are mostly hydrocarbons, or they're fatty acids, which are mostly hydrocarbons. And so what does it mean to be a saturated fat? The saturated fat is saturated with hydrogens. What does that mean? That means that that fat is holding as many hydrogens as it possibly can. What's it mean to be saturated? To be saturated means you've got as much as you can possibly hold. And so if you look closely at these molecules, you see they don't have any double bonds. And so that means that they have as many hydrogens as they possibly can, hence a saturated fat. The saturated fat, because it doesn't have a double bond, the hydrocarbons, the fatty acids, are all nice and straight. So I'm going to back up a smidge here, and again, take a look at that diagram of this saturated fatty acid. All those fatty acids are straight, which means that they can pack together tightly. And if you can pack together tightly, then you can be a solid at low temperatures. At room temperature, you can be a solid, or excuse me, at room temperature, you can be a solid because you can pack tightly together and form a solid. Now, if you look at that unsaturated fat, you see that one of its fatty acids has got a little crook in it. It's got a little bend in it. And because it's got that little bend, they can't pack as tightly. And since they can't pack as tightly, they tend to be in a liquid form at room temperature. Well, why does it have that little crook? It's because there's a little double bond in that one fatty acid, right? And so that little double bond, again, small change at the molecular level has a big change at the organismal level, or at higher levels. And so that double bond gives that fatty acid a little crook. Well, that double bond means you are not saturated with hydrogens. And so an unsaturated fat does not have as many hydrogens as it theoretically could. That double bond, you could break that double bond, and then you could attach hydrogens to each of those carbons, and then you would be saturated, right? But it doesn't. It's unsaturated. And so when we talk about saturated versus unsaturated, we mean it's saturated with as many hydrogens as it possibly can, or it's not. It doesn't have as many hydrogens as it possibly can. But again, that makes a difference in the physical properties. And so you're kind of familiar with things like saturated fats because they're considered unhealthy. And this is a tough word. There's, you talk about good fats and bad fats. That's a really bad, loose term, right? Because all these different molecules play a part in your body at some level. But some can be less healthy than others, and that maybe is a better term than good and bad. Is not a good term when you're talking about nutrition, but healthy versus unhealthy. But again, it's all relative to your physiology. At any rate, in general, a diet rich in saturated fats has been linked to cardiovascular disease, which is the #1 killer here in the United States. And these saturated fats tend to leave plaque deposits, and so they tend to be sticky. And when you have bends in your arteries and that, they tend to stick. And then when they stick, then other ones stick to them, and you get these deposits, and you narrow those arteries, and it starts to choke off the blood supply to your heart, and your heart starts to starve for oxygen, and cells start to die, and that's a heart attack. Now, there's a process in food science called hydrogenation. All right, and it's what it sounds like. It's adding hydrogen. And so it's taking an unsaturated fat. Remember, unsaturated fats do not have as many hydrogens as they theoretically could. And so what we're doing is we're saying, well, let's add those hydrogens. Let's hydrogenate this unsaturated fats. And so we're going to add hydrogens to turn an unsaturated fat into a saturated fat. Well, why would you do this? If we're saying that saturated fats might be less healthy for you, but they're solid at room temperature. And so if you wanted something like a vegetable spread or something that was solid, that had a better texture at room temperature, that's why you would do this. Or there's lots of reasons why you would do this in food sciences. Now, sometimes hydrogenating vegetable oils doesn't create saturated fats, it creates unsaturated fats. And so sometimes a byproduct of this product is something with a trans double bond. So it's, maybe you've had, several double bonds and you added hydrogens to some of them, but not all of them. Or, there's lots of ways this could happen. The point is that the process of hydrogenation creates these double bonds that are called trans double bonds. So you still have an unsaturated fat, right? If you've got a double bond, it's unsaturated. That double bond means you don't have all the hydrogens you could hold. Well, that trans double bond, the fat with a trans double bond, we also call trans fats. And you've probably heard of trans fats. Trans fats have also been linked to cardiovascular disease. And so if you've ever wondered what is a trans fat, well, this is what a trans fat is. It's an unsaturated fat that has a trans double bond. Well, what is a trans double bond? Here's a couple of examples of a cis double bond and a trans double bond. So a cis double bond, look at those highlighted hydrogens, the ones in yellow. You see they're on the same side. Cis means same. And so if you have a double bond with the hydrogens on the same side of the double bond, that's a cis double bond, and it's not as tightly linked to cardiovascular disease. But if you produce this trans double bond where you've got a hydrogen on one side and then a hydrogen on the other side of the double bond, that is a trans fat, and that has been linked to cardiovascular disease. Again, what is my theme? A very small difference at the molecular level has a big result at the organismal level. This trans double bond, the only difference is where one of these hydrogens is. But that's the difference between being linked to cardiovascular disease and not being linked to cardiovascular disease. So again, that's my theme. But you've probably heard of these trans double bonds and maybe you wonder what they were, or these trans fats and you wonder what they were. Well, this is where you get that term. Okay. There are some fats that are more healthy, or that are linked with better health. And so there are some unsaturated fatty acids that are not synthesized in your body. You need them, but you can't synthesize them. So you've got to get them in your diet. You have to eat things that already contain these unsaturated fatty acids. And so whenever we have that in nutrition, they call that an essential nutrient. It's something that your body can't produce, so you've got to get it from your diet. One type of essential fatty acid that you might be familiar with and that you've heard of are the omega-3 fatty acids. So you've heard of this, right? You've heard of things that are high in omega-3 fatty acids, and you need to get more omega-3 fatty acids. My doctor's got me taking fish oil pills, and so I'm taking extra omega-3 fatty acids. These things are important for growth. They provide protection against cardiovascular disease in developing, babies, and that omega-3 fatty acids are really important for like neural growth and things like that. And so that's why they're important, because your body can't synthesize them, so you have to get them from the diet. So another quick quiz yourself. If you've heard of omega-3 fatty acids, where do you get them from? What types of food? These are commonly found in things like fish and also in nuts. And so this is one of the reasons why we encourage people to eat more fish. Americans don't eat a lot of fish. I'm a fish guy, I'm very biased, but I'm telling you, you need more fish in your diet, right? It's high in protein, it's low in carbohydrate, it's low in fat, it's high in these omega-3 fatty acids. Baked fish is really good for you. Down here in the South, we eat a lot of fish, but we fry it. It kind of negates, the benefits from it, because you're getting more of those saturated trans fats and things, more of the fats that are less healthy for you. Anyway, So that's the first type of lipid that we wanted to talk about, the fats. The next type of lipid we're going to talk about are the phospholipids, and these are also very important in living organisms. In a phospholipid, you've got a similar structure to a fat. You've got a glycerol, so you've got that 3-carbon molecule, and on 2 of the carbons you have the fatty acids, just like in a fat, but the 3rd carbon, you replace that fatty acid with a phosphate. And so there you get a phospholipid. It's a lipid that's got phosphate in it. Now, recall that those fatty acids are made of hydrocarbons. And hydrocarbons are hydrophobic. They don't like water. So in a phospholipid, you've got those 2 fatty acid tails that are hydrophobic, but the phosphate group is hydrophilic. Phosphate likes to interact with water. And so what you end up with in a phospholipid is a molecule that's got polarity again. It's got a hydrophobic end, where the tails are, and a hydrophilic end, where the phosphate is. And so here's a couple different ways to draw a phospholipid. And so you can, again, on the left, you can look there, you can see you've got the 3 carbons, and on 2 of those carbons you have those hydrocarbon tails coming off, and that makes the hydrophobic end. And on the 3rd carbon, you've got that phosphate attached. A phosphate is a common molecule that we see. It's a phosphorus with 4 oxygens. And again, you know, if you look at that phosphate, you see one of those oxygens has got a negative charge. You remember that water is polar. It's got a negative and a positive end. So you've got this phosphate with this negative end, which interacts with the positive end of the waters. That's why that end is hydrophilic. And again, if you look at this, you see how we just took a fat, like we were just talking about, and we removed one of those tails and replaced it with a phosphate. That's how we made a phospholipid. Now, the phospholipids are mostly important because they form the membranes around cells. And again, you may be familiar with this. The phospholipid bilayer is how you get a membrane around all cells. Okay? And so, by understanding the properties of these phospholipids, you can understand how you can form something like a cell membrane. Because we've got, all these phospholipids, and we mix them in water, right? And, all living things are full of water, and the cells are an aqueous environment. Well, what's going to happen? Those tails are hydrophobic. They don't want to interact with the water. The head, where the phosphate is hydrophilic. It wants to interact with the water. So what happens is you form a bilayer, 2 layers of these phospholipids, where the hydrophobic tails are all tucked in to the inside of the membrane, and the hydrophilic heads are all pointing out. And so you've got water, then you've got the hydrophilic head of one layer, then you've got hydrophobic tails, and then you've got the hydrophilic head of the other layer, and then more water. So, by mixing a bunch of these together, they spontaneously form this phospholipid bilayer. This is how you get a cell membrane. And when we talk about cells, we're going to talk about this in a lot more detail. Here's the figure from your book. Again, just showing another way to draw this phospholipid. And you can see the 3-carbon molecule, and 2 of the carbons have fatty acids, 1 of the carbons has a phosphate. Okay. The last type of lipid we want to talk about are steroids. This is another important class of biological molecules. These are lipids, right? So they're got a lot of the same properties as the fats and the phospholipids. But what makes them unique is that they form rings. And so what happens is those hydrophobic tails, or those tails that come off a glycerol, form a ring. And so then these rings fuse together, and so you've got 4 rings, and that makes up your steroid. And so we're going to take a cuteness timeout because somebody just wandered by. So everybody say hi. Hey, Rosie. Come here, sugar bear. Come here, sugar bear. Come here, sugar bear. This is the advantage of being outside and being home, because you get a cuteness break. And this is Rosie, and she's a Cavalier, a King Charles Cavalier Spaniel, and she's very sweet, and she came by to say hi and say hi to everybody. Hello, everybody. Okay, so you go back inside, sweetie. All right, where was I? Her cuteness overwhelmed me, but now she's not listening. She's not going inside. Oh, there she goes. She's a good girl. Okay, anyway, back to steroids. So we've got these 4 fused rings, and so here's a very common steroid, cholesterol. Again, cholesterol is something that you've heard of. You're familiar with cholesterol in your body, and cholesterol is a steroid. And so you can see the 4 rings here. You've got 3 6-carbon rings, 1 5-carbon ring, and then you've got a few other carbons and a few functional groups, but that's the general structure. And so, like I said, this looks at first very different than those other lipids, right? Those other lipids had those long hydrocarbon tails, but what happened is those hydrocarbon tails just formed rings, and the rings fused together, and so that's why these are still classified as a lipid. Now, cholesterol, again, you're familiar with, right? And a lot of times you're familiar with cholesterol because a high cholesterol diet has been linked to cardiovascular disease. But this is why, again, we don't say certain things are good for you or bad for you. That's not a good term to use when you're talking about nutrition, because you could say, oh, cholesterol, it's bad for you. Yes, lots of cholesterol tends to be unhealthy. However, you need cholesterol. Cholesterol is very important for the cell membrane. It helps to keep fluidity in the cell membrane. So you have to have cholesterol in your body. It's just you don't want too much. So, that's why we don't like to say good or bad. We say healthy or unhealthy, or more importantly, we just understand the importance of these molecules in your body. Steroids are also important in your body because they're used as hormones. And so later on, we'll talk about the endocrine system. Hormones are basically just chemical signals. And so there's lots of ways your body can pass signals around. Your nerves are very good at passing signals, right? But hormones are also a way to pass a signal. And so you dump these chemicals into your blood, they travel around your body, and they go act somewhere. That's a way to pass a signal. That's what a hormone is. Well, there are lots of different hormones and lots of different types of molecules that can be used as hormones. Steroids are important lipid hormones. Now, we've seen these before in an earlier lecture. We saw estradiol and testosterone, the female sex hormone, the male sex hormone. And these do different things in your body, but they are steroids. You can see the 4 fused rings there. And so that's the type of molecule they are. They're a steroid, which is a type of lipid. One last thing that I want to bring up, and again, these are terms that you've heard before, and so we're going to try and study them a little deeper. You've heard of LDLs and HDLs. And again, a lot of times when we're talking about cholesterol, and we're talking about levels in your blood and things that are linked to good health or poor health, LDL stands for low density lipoprotein, while HDL stands for high density lipoprotein. And so what's a lipoprotein? It's exactly what it sounds like, a lipid and a protein joined together. So why are LDL, whenever you get a blood test, your doctor a lot of times will check your LDL level, your HDL level, along with your triglycerides, along with your cholesterol levels. All of these things have been linked one way or another to mostly cardiovascular health. Why are these important? Why do we care about LDLs and HDLs? Both of them are important for carrying cholesterol around the body. Remember, you have to have some cholesterol in your blood, or in your body. You have to have some cholesterol in your diet. Your body needs cholesterol, right? And so that cholesterol needs to be carried through the blood around your body. That's what LDLs, one function of LDLs and HDLs is to bind to cholesterol and carry it around your body. Now, HDLs are smaller. how do I know that? Because they're high density lipoproteins, right? High density means you can get a lot in a small area or in a small volume. How do you get a lot of things in a small volume? If they are small. So if you have small molecules, you can pack a lot of them into a volume, and so you can have a high density. That's a high density lipoprotein. Well, these small molecules carry cholesterol, and they carry cholesterol to your liver, where your liver can break it down and excrete it. And so if you have too much cholesterol, the HDLs will carry that to your liver and you can get rid of it. And that's a good thing. You don't want too much cholesterol because it's linked with heart disease. The HDLs, because they're small, they can move around your blood and make it to your liver and excrete the cholesterol. The LDLs are larger. They're low density. You can't pack as many into the same space because they're bigger molecules. They're also carrying cholesterol. They also would carry cholesterol to your liver where you can get rid of it, but they can't because they're too big. So these LDLs bind cholesterol. Because it's a bigger molecule, it tends to not move as well through your blood, so it tends to get stuck in your blood vessels. And that's one way that you can form these plaque deposits. And so again, just by understanding the molecule and its size and its properties, you can start to understand a little bit about what it does in your body. Okay, so LDL and HDL, these are lipoproteins, a lipid, which we just talked about, plus a protein. Well, what's a protein? Well, you're in luck, because our next lecture is going to describe proteins. So let me know if you got any questions, and I'll see you in a little bit, and we'll talk proteins. Later.